right, so I will give you a general overview of how this terrible virus infects our cells. Can you see my screen?
Yes.
Yes, very good. Okay, let's go. And in doing so, in telling you how this nasty virus enters our cells, I will give you a glimpse of how we use imaging techniques, mostly automated fluorescent microscopy of all kinds and automated image analysis to get all the information we need from our experiments. So without further ado, you all know by now that we are within still a pandemic. In 2019, a new coronavirus called SARS coronavirus 2 was discovered, apparently from China. But you remember that in 2002, there was the first time that you heard the word SARS. And that was the first SARS virus, which lasted basically 1 year. It infected only about 8,000 people, of which 774 died. And then after 1 year, it disappeared. The two of them are extremely similar. The two viruses are so similar that they even use the same receptor, which is called ACE2. So to attach to our cells, these viruses use the same antenna, okay? Receptors on the surface of our cells, particularly in your nose and in your throat. The cells have these receptors, they bind there, and then they deliver their genome. But here is the epidemic updates. As of today, 544 million cases, 6.3 million have died, at least with coronavirus. And then the question that we wanted to ask is, why is this second virus so infectious if it's so similar to the previous one, which only lasted 1 year and didn't infect nearly as many people? So the first thing you should know, and probably you already know because this has been discussed many times in the news, is that the old coronavirus, the SARS-1, it mostly infects your lung cells, but it's not really good at replicating and multiplying in the upper respiratory tract, okay? Whereas the new one is extremely efficient at infecting the cells of the nose and of the pharynx, so the upper part of your respiratory tract. And what is the difference of this? Well, the difference is that the new virus is released from an infected person when you are just breathing and talking. Imagine the difference. When you have SARS-1, which is the virus in green here, it infects your lungs. So in order to infect another person, you need to have symptoms. You need to cough really, really strongly or sneeze, and this virus has to come out from your lungs. But the new one, in 80% of the cases, doesn't even get any symptoms. So in other words, you are spreading it without even knowing that you have it. And what I wanted to understand back in 2020 was what gives this new virus the ability to infect so well the cells of the upper respiratory tract, and why was the old one not able to? Because I thought that this will give me really the key knowledge to understand how should we block this new virus and why is it so dangerous. And so to cut the story short, when we zoom in into the atomic structure of the surface of this virus, by now you've heard the word spike many times. Spikes are these little towers here that stick out of the virus. It's a little bit like the sea urchin, you know, this round sphere with all the spikes coming out, and there are these spikes that bind to your receptors. And you can see here in the very high resolution atomic structure of this complex spike, there is a portion that it's bound to the rest of the virus, which is made of lipids. This virus is like a cell. It's made of lipids. Inside these lipids, there is the genome, and outside there are these protrusions that attach to your cells. And specifically, this portion here in green of the spike, it's called the receptor binding domain, RDB. It binds to ACE2, but then the new coronavirus has an extra key. a little portion that sticks out of the spike, which is not present in the old virus. So the first SARS does not have this second key. It can only bind to one receptor, whereas the second one, we found that it can bind at least to another receptor, which is called neuropilin-1. And when the cells have both, then the infection grows stronger. So in a three-dimensional model that we can create using atomic structures available, we can imagine what is happening. So if a virus here arrives, this is the membrane of the virus on top of the image, and the spike in red, it arrives in your nose, it binds your receptors in the nasal cavity, in the cells of your nasal cavity. This will be the membrane of your cells. It binds to the first receptor, and then if these cells have another receptor called neuropilin-1, then the second part binds to the neuropilin-1. Now the infection is more violent, it's more effective, all right? And this second key was absent from the first SARS. So this at least explains in part one more weapon that this new virus has, and we identified what the second key binds to. So now we can imagine how we can block the virus by blocking both of these access points. And the most interesting thing of these studies, which we then published back in 2020 and wrote some reviews, I wrote some references here if you want to read more about it. But the interesting part is that if SARS-CoV-2, the virus that causes the COVID-19, has a surface protein, the spike, with basically the ability to bind to not only one receptor, but two, and this is thanks to this little extra portion here that it's in magenta color that we called sender sequence for C-end rule sequence. The same sequence is present on the surface proteins of the most pathogenic human viruses. They all have the same. And for many of them, we and others have confirmed that not only they bind their first receptor, they also bind these neuropilins. So it almost appears as in order to effectively infect and spread in the human body, a virus doesn't not only need one receptor, but somehow it has to evolve to bind a second one, which we identified being neuropilin-1. When the cells have both, then the infection spreads from one tissue to another, and the virus becomes more dangerous. So that's one part of the study that we are focusing on understanding. Is it really so that by blocking this interaction between a virus and a co-receptor, do we make the infection less dangerous for all of these viruses? We will soon see. But for the talk of today, I would like to give you an overview, starting from a simple scheme of how many strategies does a virus have to infect your cells. And when I say strategy to infect the cells, what I actually mean is what kind of mechanism allow a virus that arrives on the surface of your cells. Imagine the white part is the empty cavity of your nose. These are the membrane of a cell in your nose. And when a virus arrives, it binds to one receptor or to two receptors. And then it has three options. Either the cell swallows the virus, thinking that it is food, and it brings it inside the digestive tract of the cell, which are the endosomes and lysosomes. And when this virus arrives in the stomach of the cell, very similarly to your stomach, there are digestive enzymes, proteases. These digestive enzymes, they start to cut the surface of the virus, and these little cuts, they cause a change in the structure of the spikes. And these changes trigger the fusion of the viral envelope with the membrane of the lysosome, so that the interior part of the virus, the genome, is now injected into the cell. And this injection can happen either from the stomach of the cell when the virus is eaten up, or if the right proteases are already present outside the cell, and if the environment here, as I will show you in a moment, is the right one, which is actually what happens in your nose. So if the chemical environment here is correct and the right protease is here, then the fusion of the viral membrane with the cell can happen at the plasma membrane. So now the virus is injecting its genome through the roof of your cells. And then the third option is when you have two receptors. In this case, the cell takes the virus in, but instead of delivering to the stomach, it brings it into another compartment where more proteases are concentrated. So the fusion event and the delivery of the genome is even more efficient than in any other pathway. Okay. I have a message in the chat, which I'm going to check. It says that if I've missed the morning sections, they are all there. Thanks a lot. Wonderful. Okay, so basically from what I've just said, really to summarize, what is that defines the ability of this coronavirus to infect you? It's basically the availability in the cells that are infected of proteases, digestive enzymes. This virus cannot infect you if it wasn't for these little proteins of yours that actually cleave the spikes in a specific places. And then this changes the conformation of this spike. They perforate your membrane and they inject the genome. Okay. So coronavirus infection is determined by the availability of host proteases. But now I would like to show you how we can follow these events at single virus resolution and even single protein resolution in live cells. What we can do using powerful microscope, actually this one we used here is called lattice light sheet microscope. It's a prototype built in Harvard by my collaborators, Tom Kirchhausen and colleagues. So this is not yet available. It's basically a light sheet microscope with the resolution in Z, which is superior to that of a confocal, about twice as good. And the ability that this microscope has is to image the entire cell within few seconds with the resolution of single virion or single molecules in some cases. So in other words, we can follow the infection throughout an entire cell or tissue at the speed of a few seconds per image throughout the whole cell. So what we can do, we can basically chemically label the spike of a virus with a fluorophore, which is magenta, as you can see here. And then the inside of the virus, the core, we can genetically engineer this virus so that it expresses a fluorescent protein, which is now schematically depicted in blue, light blue. And so when a virus arrives to the surface of the cell, you see the two colors, the magenta and the blue, coincide. They create a white spot. But few seconds later, about 10 seconds later, then the virus has fused to the plasma membrane of the cell and releases the core that you now can see detaches in few seconds later and goes deep inside the cell. Whereas the rest of the virus, the spike, they diffuse laterally, as you can see here, until they disappear. So basically the rest of the virus is now become the same as the surface of your cell, and the spike, they diffuse away. So we can see a single virion arriving on top of your cell, delivering its content, basically, which means that you are now infected. And we can track this quantitatively in thousands and thousands of events in millions of cells very quickly. So imagine now we can try different drugs, we can try different conditions, and it is thanks to this technique that we realize that if the pH of the cells outside is below 6.8, so a little bit acidic, then the fusion can happen. But if the pH is neutral, for example, above 7, then as you can see in this time frame, nothing happens. These viruses sit on the cell, even if the right proteases are here and they get cleaved, but they cannot fuse, they cannot deliver the content into the cell. So it is really crucial that the pH of the environment should be below 7. And here my question immediately was, okay, this is in the laboratory. What is the pH of my nose? Do you know what is the pH of your nose? Well, in collaboration with the hospital, I convinced enough people to measure the pH of their nose. And we did it with some catheters, pH sensors that you can stick inside your nose and then measure the temperature and calibrate everything very accurately. And basically in the left and right nostrils of your nose, the pH is about 6.6. So basically, this virus has evolved to be extremely efficient at infecting your cells within seconds, even without needing the stomach of the cell, because the pH of your mucosa is already acidic. And now, actually, it's interesting to ask these two people here that have pH in their nose above 7, if they ever got infected, because maybe they haven't. So perhaps fighting coronaviruses should be as simple as eating a lot of salmiakki that release ammonia, and then they will probably stop it. Would be interesting to test. Of course, we can also observe the release of the content of the virus in the cells where you don't have these proteases here. The pH is not acidic, for example, cells of your lungs. Then the virus needs to be inside and reach an acidic compartment, which is the stomach of the cell, the lysosome. Then the right proteases is here, and then the fusion happens and the genome is injected. And now you can see a virus arriving on the surface of the cell, goes inside, and then only about 15-20 minutes later, you start to see the delivery of the gene. In this image, I didn't show you the image of the lysosome, but we also have a third color that shows the organelle from where this fusion happens. So now, beautiful, now we can see basically a single virus and how it falls apart and delivers the genome, part of the proteins. We can follow every step. But the technical advancement allowed us to basically move into literally the cells of our nose. So what I was able to do now, finally, after obtaining all the ethical permits that are required to do this, is to develop a very non-invasive technique, a very gentle technique. This was done with rhinologists at the Hospital of Helsinki. So a technique where we use a very small brush, like very similar to the one you use to, wash your bottles, your dishes. But this one is tiny, and you brush the interior of your turbinates in your nose. You collect 1 million cells per nostril. You grow them in the lab. Then with the right ingredients, the cell starts to differentiate, and then you do something that any cell biologist would consider horrible. You basically remove the liquid from the top of the cell, and you only leave it on the basal chamber of these multi-well chambers here. So what happens is that here the cells are sitting on a membrane, and this membrane has little pores. So actually the cells can eat from the bottom, but they only have air. And when that happens, for reasons that I don't completely understand, it's a complex differentiation mechanism, but the cell, they differentiate exactly like the cells in your nose. And when they're exposed to the air, they produce mucus, they produce the cilia that starts to move the mucus, and you can perform infection experiment into this three-dimensional tissue that we recreate in the lab within a month, starting from our own cells. And this is possible now thanks to the availability of very compact, ultra-fast spinning disc confocal microscope. We were very lucky to obtain one from Yokogawa. This is called the Cell Voyager CQ1. It's an amazing microscope. It fits inside the laminar hood. It's that small. It can image for thousands and thousands of hours and images three-dimensional tissues in 96-well plate completely automatically. So there's no binocular. You don't look for the cells. The microscope analyzes them all. If you are interested, I have more slides later on. But for the time being, now we can recreate the tissue of our nose in the laboratory. And we can study how the cilia that are exposed on the surface of the cell, they capture viruses, and then the viruses go inside the cell. And we can see the cytoskeleton around it. So the skeleton of the cell, which is in yellow. We can see the nuclei underneath. And we can see the presence of the famous protein neuropilin-1 in many of these cells. Actually, these are cells from my nose, if you're curious, who is the donor. So I have a lot of neuropilin, might explain why I've got this virus already twice, regardless of my vaccines. So I'm a very good host for coronaviruses, apparently. And this brings me basically to the second part of my talk. So now basically from the ability to follow a single virus and see how many mechanisms it has to enter the cells and what are the cellular factors that are required to allow this mechanism, we can come up with a combination of drugs, with a cocktail that blocks every possible access route. So we try to block infection by blocking all the entry routes to the cells. And to do this, to see if it works, we can do an experiment in mice where we give a couple of drops in the nose of the mouse of this medicine, and then we infect the mouse and see if there's infection in the lung. Okay, so here I have to already thank a wonderful set of collaborators, particularly in the laboratory of Olliva Palatti at the Faculty of Medicine and Helsinki Hospital, Ravikant, Lauri, Suvi, Teemu, and Thomas, because without them, we would have not been able to do all this experiment in such a short time. But the ability that we have to access now tissues from humans, mice, isolates viruses from people and sequence them, it basically allows us to formulate potential cures that could be delivered with a nose spray, for example. So imagine you are home, one of your family members is positive, then the whole family takes a couple of shots per day for a couple of days, and then you might be cured. So to test if this works, we gave really 2 shots per day in the nose of these mice, which received a very high dose of virus. And then 2 days later, we see, can we measure viral replication in the lungs of this virus? Is there any virus left? So we can use PCR from the lungs of the mice that we sacrifice. And here's very quickly the results of this experiment. If you don't put any virus and you analyze the lungs, you don't get any PCR values. Okay, so this counts basically how much virus genomes you have in the lungs of these mice. No virus, no signal. If you infect the mice, but you don't put no drugs, then all of these mice have a lot of virus in their lungs. Okay, now if you put only one of the drugs, but not the other, you don't really do much. Actually, some mice, they even have more infection. If you put the other drug, the drug number 1, there is a decrease, but as you can see, it's not enough to completely remove the infection from your lungs, which is not good, because imagine if you allow a virus to learn what to do in the presence of a drug, you can imagine sooner or later that could be a resistant mutant, or anyway, when you stop the treatment, the infection can spike again. So we really wanted to block it completely. So we start to combine the drugs at decreasing concentration, and we are able to completely erase the infection from the lungs. Like unbelievably strong results. The mice are fine, their lungs are clear, they look happy, and they live ever after.
Giuseppe, 4 minutes to go.
4 minutes to go. I have the last few slides. Perfect.
Okay, good.
I made it this time. Thank you. So, okay, now this experiment is promising because it tells you that if you take a medicine in advance, you can protect yourself from a coronavirus completely. There will be no infection in the lungs. But what happens if you already got it? If you go to the movies, then in the train station, you get the infection, the test is positive. Does this drug help afterwards? So we did another experiment. First, we infected the mice for 6 hours, and then we gave the medicine for 2 days, twice a day, okay? And then we do an autopsy, and we take a slice from the lung. This is the slice from the lung of these mice. And whenever you see brown, this means that the cells of the lung are infected. So the brown color is an histochemical staining for the viral protein, nucleoprotein. Okay, so it means these lungs are full of virus. If you put no drugs, then the virus not only infects you, but if you use an antibody to see if there is cell death in the lungs that are infected, you see a lot of cells are dying. And these are exactly the cells that are infected. Okay, so this is another antibody that recognizes a protein which is a marker of cell death by apoptosis, cleaved caspase 3. So infected lung, a lot of cell death. But now we put the drug 6 hours after infection. So you do see that there are some cells infected in the lungs, but there is no trace of cell death. In fact, all the cells are completely clear, completely blue. So in other words, if you take these medicines that basically block all the entry routes of this virus. You take it before, you prevent the infection. But if you give it few hours after you've got the positive results, then you can still protect yourself from damage. So it can be a prophylactic treatment, but it can also cure you afterwards and help with the symptoms. With that, I will stop here because I had only limited time. So I just wanted to give you a glimpse of how basic research and powerful microscopy could give us the knowledge we need to pick up the right drugs that eventually can become really a cure within a couple of months of studying. And with that, I have to thank the people from my laboratory that are involved in this particular project. So it's Ravi, Angelina, and Tuukka, who is our lab manager. Then the group of Olliva Palatti, particularly Ravi, Lauri, and Suvi. People from the hospital, they helped me with taking cells from the nose, analyzing pH of people, and so on and so forth. So all these experiments are done by real doctors. Then our pathologists that tell us if the mice are doing well or not, how do their lungs look like. That's Anja Kieper, University of Zurich. And then Thomas and Teemu. Thomas is a group leader nearby my lab. He does basically the PCR with his students, and Thomas does the sequencing of the viruses to make sure that we are really working with correct viruses. And thank you all for your attention, but particularly I would like to thank the Yokogawa company, Mahomi, Malin, Daniel, Arvon, Gerd, David, and Mikael Schell, who basically gave us a high throughput, ultra compact spinning disk confocal microscope to help our research in COVID-19 for 2 years now. And without them, I would have not been able to do anything. Of course, we have fundings, which I have to acknowledge all the time. So the European Union, University of Helsinki, Academy of Finland, and the Australian government for funding our research. And thank you all for your attention, and I'll be happy to take some questions if there are any. Thank you.